Okay, so in lecture one, we went over the race to determine which molecule was responsible for the passing of hereditary information from generation to generation. Um, After concluding that it was DNA, not RNA or proteins, the race then came to determine what the structure of the DNA molecule looked like. And remember that there were certain criteria that needed to be met in order to have DNA be that molecule. And what we saw was in the 1950s, Watson and Crick built the molecule and showed the complementary base pairing, how information could be stored in the molecule itself, and also how the molecule can be passed on from generation to generation, and also DNA replication, so how the molecule could replicate itself. And that's what we're going to focus on next, DNA replication, and then looking at going from DNA to making proteins. So in replication of DNA, DNA replication, which you know occurs during interphase of cell division, the DNA will replicate itself, is the process of copying a DNA molecule. And ultimately what we say is that this process is a semi-conservative type of process in that each strand of the original double helix serves as a parental strand or a template to build a new strand, or what we call a new daughter molecule. So if we look here we could see representative of the semi-conservativeness of DNA replication. The blue strands will represent the parental strands, and the green strands will represent the newly synthesized daughter strands. The building of the new strand is from the parental strand, which serves as a template or a blueprint to build that new strand, following the rules for complementary based pairing. Remember that DNA always occurs, replication always occurs in the 5' to 3' direction. So, the mechanism of replication of DNA. Replication requires the following steps. First, there is the unwinding or the separation of the two strands of the parental DNA molecule by an enzyme called DNA helicase. Single-stranded binding proteins, or abbreviated SSB, are going to attach to newly separated DNA and prevent the helix from reforming. Following the rules of complementary base pairing, remember A with T and C with G, between nucleotide and nucleotide on a template strand, the DNA primase places a short primer on the strands to be replicated. So DNA primase is the primer that is going to indicate where replication should begin. DNA polymerase recognizes RNA and begins DNA synthesis. So the two strands are replicated differently. One strand will be called the leading strand. The other strand will be called the lagging strand. And that basically is in association with the way replication occurs in that 5' to 3' direction. The leading strand will be replicated on a continuous basis, and the lagging strand will be replicated in small fragments called Okazaki fragments. And you'll see that in a minute. The joining of the nucleotides in the lagging strand then is done by an enzyme called DNA ligase. DNA ligase will form that new strand so that each daughter DNA molecule contains one old and one new strand. Replication in eukaryotes occurs in thousands of places. In prokaryotes, it's a little different. Prokaryotes, remember that in the prokaryotic genome, the DNA is often in a circular loop of DNA called a plasmid. So bacteria have a single circular loop of DNA stored in the nucleoid, and the replication moves around the circular DNA molecule in two directions. It produces two identical circles. The process begins at a place called the origin of replication and will proceed until the entire chromosome has been copied. in essence, that's about 40 minutes. But the cell divides every 20 minutes. So a new round of replication can begin before the previous round is actually completed. So it's pretty magnificent thinking of it in the prokaryotes, in bacteria cells. And if you look at prokaryotes, you could see that when we talk about bacteria cell dividing every 20 minutes, and we look at, in our world today, the battle we have with drug resistance, of antibiotic drug resistance with bacteria, remember that during every time DNA replicates, it allows for some degree of mutation. So each time bacteria will replicate, you get some mutation into that DNA, and some of that mutation could be drug-resistant mutations, so that bacteria over time, every time it divides, is going to now pass on that specific trait to the new bacteria cell, ultimately leading to a population of bacteria now that are drug resistant, such as MRSA, which is methicillin-resistant Staphylococcus aureus. Eukaryotic replication begins at numerous points along each linear chromosome. The DNA unwinds and unzips into two strands. Each old strand of DNA is going to serve as the template for the new strand, and following the rules of complementary base pairs, the new strand will be formed. This requires the enzyme called DNA polymerase. So, eukaryotic replication occurs at an area called the replication bubbles. Replication bubbles spread bidirectionally until they meet. The complementary nucleotides are joined to form the new strands. Replication is a semi-conservative process. One original strand is conserved in each daughter molecule. Each double helix has one parental strand and one newly synthesized daughter strand. Polymerase is unable to replicate the ends of the chromosomes. Remember that the ends of the chromosomes are called telomeres. So telomeres are then copied by an enzyme called telomerase. Unregulated telomerase activity negatively affects the cell function. So if we look, here we have prokaryotic replication. Down here we have eukaryotic replication. So you can see the difference between the two. So if we look, remember that there is one strand called the leading strand. The other strand is called the lagging strand. Here is the leading strand that is being replicated. So DNA helicase is going down the parental strand of DNA and it's unwinding it. And that ultimately leads to this replication bubble. Following behind DNA helicase is DNA polymerase. DNA polymerase is adding new nucleotides to build a new strand, the new daughter strand, based off the nucleotides or the template in the old parental strand. This happens continuously in the 5' to 3' direction. However, remember that the strands run anti-parallel to one another, all right? So this strand, it can't run continuously. So what happens is, as the DNA molecule unwinds or opens up, you expose the nucleotides in segments. So DNA polymerase will come in and it will add new DNA nucleotides following the rules of base pairing. But then it eventually will get to a point where it already has replicated that strand. So then it will have to come back, and as DNA helicase unwinds more and exposes more nucleotides, then it will come back and build in more strands. And then as DNA helicase unwinds even more, it will come back and even put in more strands. So those strands, or those segments of newly replicated DNA, are called the Okazaki fragments. And eventually what will happen is you need to fuse all those fragments together. That is where DNA ligase comes into play. So DNA ligase will fuse all those strands together. So the accuracy of replication, DNA polymerase is very accurate, yet makes a mistake about once per 100,000 base pairs, and it's capable of identifying and correcting those errors. Now, as one would age, that of course becomes more significant. So, those are the enzymes that are involved with DNA replication. And that is the process of DNA replication. I will go over this maybe a little bit more in class. If you have questions, please feel free to ask them. The genetic code of life. Basically what we see is genes specify enzymes and other proteins. And that a couple of scientists that really played a significant role in the whole idea of this concept. One of those guys is the name of Archibald Garrod. He described inborn errors of metabolism. And then really important is the work done by Beadle and Tatum. And Beadle and Tatum, basically what they looked at is experiments on a fungus called Neurospora crassa. And he proposed that each gene specifies the synthesis of one enzyme. So genes are specific to proteins. Each gene will specify a protein. In this case, that protein would happen to be an enzyme protein. So one gene, one enzyme hypothesis. And ultimately, the flow of genetic information is from DNA to RNA to protein to an observed trait. So the genetic code of life. The mechanism of gene expression. DNA and genes specify information, but information is not structure and function. Genetic information is expressed into the structure and function through protein synthesis. The expression of genetic information into structure and function ultimately is this, twofold. DNA in a gene determines the sequence of nucleotides in an RNA molecule. And the RNA controls the primary structure of the protein. So the sequence of bases in the RNA are going to code for the amino acid sequence in the protein. So here you could see when you look at RNA, remember that RNA is a single-stranded molecule, and it has three similar bases to that of DNA, with the exception of uracil is going to replace thymine. And the 5-carbon sugar here is ribose and not deoxyribose. So the central dogma of molecular biology is what we are going to look at now. So this is like the whole thing of molecular biology. It comes down to transcription and translation, going from DNA to messenger RNA, and then messenger RNA into the amino acid sequence of a polypeptide and protein. And we know that when we look at that, one of the strands of DNA is going to be transcribed in the messenger RNA. That strand would be known as your template strand. And then the messenger RNA is going to be read in triplet code called codons. So a codon is 3 nucleotide base pairs read together to determine which amino acid will be coming into play. So ultimately what we see here is in translation then, that messenger RNA, using the help of 2 other RNA molecules, tRNA and rRNA, are going to determine the polypeptide or the amino acid sequence of that polypeptide. And in this case, we have serine, aspartate, and proline that are all coming together there to form the building polypeptide. This ultimately is called protein synthesis. And we'll continue with protein synthesis in our next lecture. That is where I'm going to stop for now. Please continue reading on in the book and work on your Chapter 12 packet. Thank you, everyone, and have a nice day.